All right, guys. We are back again to look at some more questions. This time we are going to use or be given some 3 specimens or 3 organisms that we're going to use to answer some questions, right? Okay, so we are instructed to study the diagrams and answer questions 2A to 2J. We've been given organism A, B, and C. All right, so let's get right into it. So with A, we've been asked to identify the organisms illustrated in diagrams A, B, and C. That's question 2A. Identify organisms illustrated in diagrams A, B, and C. So identify means to name. So we are just naming them. So A, looking at the shape, the irregular shape, formation of extensions of the body parts here and there. It tells you directly that it is amoeba. Okay, it's amoeba. So amoeba is fine. You have different types of amoeba, but you can just go with amoeba. Start writing with a capital letter A. And then looking at B is Euglena. Then C, looking at the fact that we have it in a filamentous form, thread-like with cells, join end to end. Then we have Spirogyra. Of course, we're also looking at the spiral chloroplast of it. That tells us that it's Spirogyra. Okay, then we have to name the parts labeled. So lots of parts have been labeled here. We have been asked to name some of them, if not all. So we have I, and so on. Okay, so for I is on A, and we are looking at a little challenging to see exactly what we have here, but it's like one part of the cytoplasm, which is the ectoplasm, all right, of the cytoplasm of the amoeba, right? So I is ectoplasm. You can see that the arrow is pointing close to the the cell membrane, okay, and not exactly within the cell, which would have given us endoplasm. So you can go for ectoplasm right here. Okay, then let's go to the right side, and then I. So I again, you can find it in the amoeba right here. So what do we have? We have an organelle which is transparent, and that tells us that it should be the contractile vacuole. So it contains water, so usually it's transparent. Water is transparent. So all right, so we have the contractile vacuole. Then we come to VI. With VI, the guideline, so we are seeing it on B. So VI, the guideline is pointing at a dark spot close to the outside of the, what does it look like, the gullet or something like that. Yeah, so that tells us that is the eye spot. All right, the eye spot or the stigma, that photosensitive or receptors on the amoeba. All right, then we have VIII. VIII looks like the nucleus of the Euglena. Is B, right? We find it in B. All right, then we come to IX, still with the B. And that's the whip. The structure for, that is whipped for movement. That's a flagellum. It's only one, so it's flagellum. Then we go to XI still with the Euglena or on the Euglena, and we have these numerous structures which can just be the but then it's not exactly pointing at the entire structure. We are looking at a core, something within that, right? Something within that can be the parinoid. So we are looking at the chloroplast, and then we are also looking at the structure within it, which is the parinoid. All right. So we go on to XIII. Where do we have XIII? So XIII, we are on C. We can find this one on C, and with this, the diagram is not all that good, but it's still good enough. You can see these structures going up and down. So that is the chloroplast, or the spiral chloroplast. It's from the is based on the nature of the chloroplast that we have Spirogyra, so spiral, so Spirogyra. Then we go to XIV. Where do you have XIV? Again with C. So what is it pointing at? It's pointing at the cell wall. It's pointing at the cell wall of C. Pointing at the cell wall. Right. Then what do we have? XV. Yes. So the outermost part, the slimy layer, is the mucilage of the Spirogyra? Yes, Spirogyra. Then XVI. So the XVI right here, we've moved away from the cell wall, and we are looking at the thin cytoplasm. The thin cytoplasm of the Spirogyra. So that's what we have. So move from the cell wall, and then we are looking at the cytoplasm. Right. Okay, so that's what we have. The answers here can be a little debatable because of the quality of the diagram anyway. So some can go for cell wall for SBI. Well, with this, it can be considered. In what forms do diagrams A, B, and C exist? Right. So A, which form does it exist? So it's a unicellular, right? It's unicellular. B is unicellular as well. And then C, we say, is filamentous, right? That's a form that it exists. C, that Spirogyra, is filamentous. Excuse me. Then we move on to D. D says, which structure in C makes contractile vacuole not so important? In C, not so important. You know that the contractile vacuole is supposed to it's supposed to osmoregulate, right? Osmoregulate, that's regulate the amount of water right? Osmoregulate. So if C has limited contractile vacuole or none, what structure will it have that makes the work of the contractile vacuole less important? And that has to do with the cell wall, right? The presence of the cell wall in Spirogyra makes the contractile vacuole less functional or less important, since the contractile vacuole can, the cell wall can allow the Spirogyra to sort of withstand the pressure that the water will impose on the cell and prevent the cell from bursting or breaking and so on and so forth. So that's it. Then we come to E, which is tabulate 3 differences between specimens A and C. So what do we have for A? So A is amoeba, C is Spirogyra. So there are 2 quite different organisms, and you can have a number of differences. You can talk about the pseudopodium in A, and present in A, pseudopodium absent in C. You can talk about the cell wall in C, which is absent in A. So A is not having the cell wall. You can talk about the food vacuole in A, a food vacuole absent in C. You can talk about the mucilage in C, mucilage absent in A. You can talk about paranoid in C, paranoid absent in A. You can talk about the spiral chloroplast in C, spiral chloroplast absent in A. You can talk about the thin strands of cytoplasm in C, which is large in A. Okay, sorry about that, battery is a little low here. All right, so what else do we have? We have you can see here the dark spot here. That's the nucleus, which is centrally placed in C. It's not in A. In A can be found almost, all over the place due to the nature of the cell that's the amoeba cell, being able to assume any shape, any form at any time. So the nucleus is likely to be almost anywhere in the amoeba at any time. All right, so what else do we have? I think we've talked about most of the differences. 2 economic importance of B. So B is Euglena. So we are looking at Euglena having chlorophyll or chloroplast, and therefore can make food. So serves as food for other organisms. And if you think about plants, and they also having that ability to photosynthesize, what else do? They release oxygen into the atmosphere for other animals to use, including plants themselves. They cause recycling maintain the natural cycles, like water, like carbon, nitrogen, carbon, water mainly. Then what else? They provide materials for production of other biomolecules, like proteins, lipids, and so on. Right. Even the nucleotides as well, or the nucleic acids. Sorry. Okay. So very important there. We go to GI. Which structures on A and B are used for locomotion? So we know that A is amoeba, so that will be pseudopodium, shown by the fin here. And then for B, that's flagellum, so it will be IX. Right here, shown by IX. So that's A and B. Then we go to H. Is it H? Yeah. State one similarity between organisms A and B. So A is amoeba, B is Euglena. So similarity, I mean, they are all cells. So we know, excuse me, that they are likely to have nucleus, yes. Cytoplasm, cell membrane will be there. So we can always go for that. Of course, the same applies to the similarity of A and C. All right, so we are almost there. What's the mode of asexual reproduction that occurs in B? So B is Euglena. So Euglena, that will be binary fission. The binary fission, right? That's the main mode of asexual reproduction. What type of sexual reproduction occur in C? So in C is spiral. So in spiral, we are likely, so we'll have conjugation, right? Conjugation in different forms. We have lateral conjugation, we have the scalariform conjugation, so on, but definitely is conjugation. All right, so yeah, I think we've done justice to all the questions. So thank you so much for turning out.